Good morning and welcome to our good news session. I have been told that this was specifically requested by all the depressed attendees at the end of last year. But we can only find 4 speakers, so that would fit the bill. This will be really interesting array here. Our order is going to be a little bit out of order from the program, but only with one little switch. Our first speaker will be Professor Tammy Hoffmann, who is speaking about improving the reporting and reproducibility of interventions with TIDIER. And I also should say that, the talk will go for about 15 minutes, and then we hope to have maybe time for a quick question in between speakers, but then we'll have more time for more Q&A at the end. So please, Tammy.
So no pressure to have some good news, but okay, here we go. So I've never heard it pronounced like that. We normally say TIDIER, but I like emphasizing the TIDIER. So a lot of health research is about interventions, the bulk of it. And so unless we know what the details of those interventions are, we lose a lot of the value in that research. So that's sort of the premise behind one of the streams of research I've done for the last 15 years or so. So if we don't have a complete description of the intervention, other researchers can't replicate or build on those research findings. And if we've got an RCT showing that an intervention is effective, unless we have the details of it, clinicians, patients, other policymakers can't actually use that intervention reliably in practice. So think of it a little bit like a recipe. You set out to make something impressive like this. If you were wanting to do this, you would need to know the ingredients and the method, what to do with them, how to combine them. If you don't, if you're missing some of ingredients or the steps in the method, what you end up producing probably won't resemble what you set out to make. So that's why when we were describing an intervention, it's something that was surprisingly overlooked for a long time until TIDIER came along, that we need to know those details. So that's what we've set out to do. What we want to know, what actually did the researchers do? What intervention did they provide? We started out by looking how much of a problem was this in trials. So in 2012, we took a sample from 2009, all the top 6 leading medical journals, so BMJ, PLOS, Lancet, etc. And we took all RCTs of non-drug interventions. And the reason we focused on that is because we knew that drug interventions from some earlier research are not terrific in their description, but they're missing less because they're easier to describe, dose, route, etc. But non-drug interventions, which is everything else, so surgery, procedures, rehab, psychosocial interventions, public health services research, there's so many elements to those interventions that they're often, they're more likely to be more incompletely reported. So we wanted to start by saying, and this is the bad news, how much of a problem is this? So we analysed all of those. There was 137 non-drug interventions in a 1-year period in those 6 journals. And we took it from the perspective of, if I was a clinician and wanting to implement this with a patient tomorrow, does it have the detail that I would need to use it with patients? So the criteria we looked at down the bottom, because TIDIER didn't exist yet, just some basic items about, what did you have to do, what materials were given to patients, what was the intensity, like how many sessions, etc., and what did you do in those sessions. And just from the descriptions in the paper or the supplementary materials or whatever we could Google and find on trial websites, we found that overall about 40% were completely described, meaning 61% were not described. We then wrote to authors and we said, if they were missing details, and we said, could you please provide us with whatever was missing? So, how many sessions were provided, or how did you actually train the clinicians? And we could salvage 20% of those trials. So we could get the information from authors. So we ended up being able to have 60% of the trials, of the interventions, would be usable in practice. But, who's going to do that? No clinician's going to have time to write to authors and ask for information. And obviously we didn't get to 100%. Some of the responses we got when we contacted authors and asked very specific questions were, Training material is not available. We had no real reason to do that. This one was common. I'm unable to find my old computer files. And this was only, 2-3 years after some of these trials were conducted. I no longer have access to the materials. That URL doesn't exist anymore. And quite a lot of authors said, no, everything you need to do the intervention is in the paper, which we disagreed with, of course. So it became apparent that if it's not in the paper, then you're going to struggle to get it down the track. And the longer it goes on, the more of a challenge that will be. But also that a lot of the authors thought what they had provided was sufficient. And I mean, why would you do a trial of an intervention? And if it's effective, presumably you're doing it because you want people to use it. So why would you not be trying to provide everything you could so it could be used in practice? But authors' awareness of what constituted a complete description, we worked out that there was some gaps there. So some folks said to us, well, surely this can't be much of a problem. There must be guidance somewhere. There must be checks and balances in the system to stop this occurring. So if we look at the existing consort guidance, item 5 is the one that talks about intervention, and it just says with sufficient details to enable replication. So clearly that's what the researchers that we contacted thought they had provided sufficient details. We took a sample of journal instructions to authors and looked at, did they give any guidance about, intervention details or materials needed? And most of those did not. Only 14% actually mentioned anything about interventions. And then they just said, describe the interventions. And then there was another study where we looked at, did peer reviewers or editors detect any missing information? And no, most of them didn't. So we thought, okay, one of the solutions to this is probably going to be to work up some guidance on how to describe an intervention, what a complete intervention looks like. So that led us to gather an international team together. I mean, Ginny was one of the members of that. So we got folks from all different disciplines together and tried to solve this problem of, if we want to describe an intervention completely, what do we need? What does that look like? So TIDIER, so stands for the template for intervention description and replication. Template meaning it's a guide, it's not completely prescriptive. There may, of course, always be more information or different information you need to provide, but it's a starting point to guide you in that process of describing your intervention. And the overall aim of TIDIER is, its name's quite self-explanatory, to improve the completeness of reporting and then ultimately the replicability of the interventions. So we worked with the consort and the spirit folk, realising there were gaps in the existing guidance. So it's an official extension of consort item 5 and spirit item 11. In a nutshell, TIDIER is the who, what, where, when, why, and how of an intervention. There are a few more items down the bottom here. So tailoring, any modifications that happen throughout the trial once it started, and 2 items about fidelity. What strategies were in place to ensure the intervention was actually delivered in the way it was intended, and then how well did that fidelity actually hold up at the end of the trial. So 12 items. Some of the feedback from folk when we piloted this was, yes, but word limit counts, so we can't provide this information. And so we explicitly say in the TIDIER paper, that's not a reason to not provide the information. And in the paper, we talk about some ways to get around this issue of perceived barrier of word count. Online supplementary materials are your best friend. Journals are very happy. I think nearly every journal now is very happy for you to provide that information. So as many online supplementary materials as you want. Put the details in the protocol. Put it in the registry. Do a video of procedure, the whole, picture speaks 1,000 words. If it's some type of surgical procedure or a rehab procedure where it's very hard to describe all the steps of the procedure, video it, put photos of the materials. So there are ways around this perceived limit of word count. So that's why in the TIDIER checklist, as well as saying where is it in the primary paper, we have a column saying, if it's not in the primary paper, please tell the reviewers or the editors where else it might be. And it might be that it's in the registry or etc. When we developed TIDIER, we were disseminating it when it was published, and we wanted to sort of overcome this, but we already do this, and why is this important? So this is just one of the little videos we used to disseminate, to try to get that message across of why this was important.
this paper, but what did they actually do in the intervention? How am I meant to know how to use this intervention with my patients? What a waste. Oh, that's such a clear description of the intervention. And there are links provided to all the materials. I have all I need to be able to use this intervention with my patients. Thanks, TIDIER.
We have a few versions of that for systematic reviews and other tools. So that was 10 years ago, and now the 10-year anniversary of TIDIER. So what's happened since then? Had 8,000 citations, so its uptake's been pretty good. It's been the subject of at least 20 editorials or commentaries in multiple health disciplines talking about why it's important to describe the intervention. Numerous journals either require it or at least endorse it in their instructions to authors. It's been translated into 7 languages by teams in those countries approaching me. And there's been a few major kind of extensions for areas, particularly where it's not. So obviously most of the TIDIER examples we use in the paper about individually delivered interventions, but something like public health and policy where you're delivering an intervention at scale, some of the examples and considerations are a little bit different. So some of the major ones has been one for public health and policy, one for describing placebo and sham controls. Now obviously TIDIER in the main paper we talk about you need to describe both the intervention from a replicability point of view and use, but also the control. We need to know what the comparator was, so that we, can work out what's additional. But when you're talking about a placebo and a sham, there's obviously other details there you need as well. And then more recently, there's been one for telehealth, and there's a few other ones around. And I'm pleased to say that next year, there's a new consort and a new spirit coming out, 2025, and TIDIER has now been integrated into consort and spirit. And part of this whole sort of reporting guideline push of not having a million extensions, which makes it clunky to use. Some of the key extensions for consort and spirit have been brought into those main papers and they'll be out next year. But of course, the problem of intervention description is not just in primary studies. We've got this domino effect. If something is badly reported in a primary study, when you then go to integrate and combine those studies in something like a systematic review or a guideline, you're missing key information in the primary study, you can't put it in the evidence synthesis, as you're well aware. So we then started looking at the issue of intervention description in interventions. Suffice to say, it wasn't good because it wasn't great. And I realize this is a business. If it wasn't great in trials, it's not going to be great in systematic review. So red means it was missing. So let's just say there's a lot of red there. So what we then set to tackle this, another group got together and we looked at how to improve the usability of systematic reviews by improving the reporting of interventions in systematic reviews. And we developed a set of recommendations there, with TIDIER being a key basis of one of the recommendations. That has been, so Matt and Joe are here, the PRISMA 2020 experts. So TIDIER is now part of PRISMA, which is fantastic, and also part of the Cochrane handbook for how to do systematic review. And we're starting to see that the Cochrane guidance, some Cochrane reviews now will have a table in one of their supplementary materials of all the details of the intervention. So if the review concludes, these interventions are effective, you can go to the table and work out what it is you need to do to implement that intervention in practice. So reporting guidelines, reporting checklists, are all well and good. The problem is at the moment, the way they're largely pitched is that it becomes the journal's problem. The burden is on the journal to be that gatekeeper and check that reporting is adequate at the time of submission, which not all journals do a great job at that. One of the bigger issues here is about how to perceive reporting checklists. I'm talking more generally here, not just about TIDIER, and moving it so that authors see it as a useful thing to do right from the outset. So at the time, and so at all these other bodies and organisations have a responsibility for encouraging this. So research funders, for example, trial registries requiring the use of, spirit if it's a trial, RCT. The authors, as I said, use it as guidance at the protocol stage, not as just some checklist you've got to fill in because the journal won't let you submit the paper unless you upload a reporting checklist. So we've got to change this kind of perception of reporting checklists as just some one more annoying piece of document to do. Actually see it as useful guidance right at the outset. And of course, there's lots of work to do in getting editors and peer reviewers to actually use it to guide their reviews and meaningfully check, you know, is this trial or whatever it is this study well reported? Is it useful? Can I critique the methods? Does it have all the information it needs? So some of the ways to do this, other than changing, well, the whole system, and I know this is good news, I'm getting there, is to think about, are there helpful tools and templates that we can integrate and provide for authors? We don't want to add more burden to researchers. Can we integrate this into existing workflows and requirements? So the Australian New Zealand Clinical Trials Registry have been an amazing example of how to do that. So very early on, about 1 year after TIDIER was published, they changed their data field, their requirement for the description, get out the way here, for the description of intervention saying, use TIDIER, follow these headings, provide this information, and they check that that's being done. So you have to do a trial registry entry anyway if you're going to register a trial, so you might as well report it completely. And that way you've either done it already to go into your protocol, or you can refer, because there's stability in trial registry entries, you can refer to that in your paper and say, you know, more details are in the trial registry entry. So it's about trying to integrate these things into existing workflows and processes. So a few months ago, we decided to have a look at this more formally. So we randomly selected 100 registry entries from the Australian New Zealand Clinical Trials Registry, 50 from 2013, because that was the year before TIDIER existed, and 50 from last year. So we had two raters independently assess the intervention description field using the TIDIER items, only 10 of the 12, because 10 of the TIDIER items are for protocols. There's 2 items about, you know, the one about was the intervention modified throughout the trial, or how was fidelity achieved? You can't actually report those till you've done the trial. So 10 of the 12 items, and now this is the good news. So in 2013, the mean out of 10 was 7 across those entries. In 2023, the mean was 8.3. So just by, I know it might seem small, but it's in the right direction. And one simple policy change, just that trial registry saying, please use TIDIER to describe your interventions, no extra effort or, you know, required, no extra process. And we've had some improvement in that. And if you want to see it graphically, you can see across the items. So the blue's 2013, orange 2023. You can see it's been across all of the items there. The other tool we developed to try to make it easier for authors, because a lot about, you know, trying to improve research reporting is making it easier, not adding to the burden, is we developed something called the TIDIER authoring tool, which is where it basically, not that as unsophisticated as you can get, but it's really just prompting the authors for each of the fields to type in, you know, what's the dose of the intervention, or, you know, what materials did we use? And then that generates a PDF that the authors can add as a supplementary material, or email to people, or upload to a registry. So it's just a nice way of presenting, sort of at a glance, this is the intervention. This is all the details, all the information you need to know how to do this intervention. So that brings us to how much of a problem is intervention reporting in trials now. Now, I was hoping to have the answer to this before today, but we haven't quite finished. So what we are in the process of doing is repeating that 2013 study we did, where we took all the trials published in those big 6 medical journals. So we are repeating that with a sample of articles, trials from last year. Two groups, one, the big 6 medical journals, but also some other journals, just so we're looking at how generalizable is it. I don't have the results. I have a sneak peek of the results. It's looking good. Here's the good news. So we've only done 19 trials so far. These are non-drug interventions. But if you remember that earlier graph I put up, well, you know, the bar graphs are a lot lower. It's looking pretty good. So watch this space. We will have the full results sometime next year. But at the moment, it's looking pretty good. And what was good to see is there's some beautiful examples. And these are in, you know, some of the bigger journals in the world. For example, this one's from the Lancet last year. There was a supplementary table with all the TIDIER items, exactly what we suggested. So that trial, people can just go to the supplementary materials, take that table and know exactly. If you're doing a systematic review, you can take that and you can integrate it. So I think there has been some improvement, certainly in awareness of the problem, the need to do something about it. And hopefully we'll have some research shortly showing that we've made a little bit of an improvement. So I think we've gone from 10 years ago, looking at papers and going, I have no idea what the researchers actually did or what the intervention was. I think we're at this stage now. I have fewer issues. We're not there yet, but I think we're heading in the right direction. So, some good news.
All right, yeah, so I interpreted, first of all, thank you very much. My name is David Mellor from the Center for Open Science. The assignment I was given was to look for some good news from meta research about what's going on in the world, and I interpreted that in a couple of different ways. So first, I was looking for examples of things getting better, not perfect, but examples of improvement out there. Next, I was looking for examples of people finding some sort of problem that was improved through policy or through other means. Other things that we've been looking for, another example of good news that I think fits the assignment for this session was a proposed policy is working as intended. And finally, look at these good things that are associated with open science practices. And I want to be very clear that this is in no way systematic. This is an exercise in looking for some good examples. And I'm happily acknowledge that this is kind of cherry picking and looking through things through a very optimistic lens. And I was actually, it put me in a good mood over the time period that I was putting this together. So thank you for the assignment. And just because I'm a little scatterbrained, these are all going to be given in different orders. So just as they come, there'll be different examples at different spaces. But let me start with some examples of areas where research is improving or where practices are improving. And the first example I wanted to showcase was work from Tom Hardwick. And actually, I'm dedicating this presentation to him because he comes up a couple of times in here looking at some examples and improvements. But him and colleagues were looking in this example of prevalence of transparency research practices. And they were comparing, this was a repeat of work that they had done in 2017. And this was an update several years later. And they were looking across several different open science practices in the psychology and the psychological science research literature across the field and in several prominent journals. And in every case that they surveyed those items, they saw some improvement, pretty modest or overlapped with the confidence intervals for materials, but particularly great improvement with data and sharing of analysis scripts. The message is a little bit more muddled with some of the other things I looked at, although conflict of interest is stood out as something that substantially improved during that time period. And depending on whether you're an optimist or a pessimist, you could say that things are either increased moderately from 2017 to 2022, but there's still a lot of room for improvement. And so depending on your perspective, that's going to be either an example of there's still a long way to go, but the trend is in the right direction. The story is similar in other fields, and when other people look at it, they see similar trends. And this is just one of several examples in biomedical research and looking for analytical code sharing. Every year they looked at it, they saw rates of code sharing increasing up to just about 50%. And it was somewhat interesting that the effect that different policies had. So they teased it apart based on journal policies that had either encouraged analytic code sharing or mandatory code sharing. You can see, a bit of a difference from those two, although it's a little bit perhaps a little bit discouraging that the mandatory code sharing is not higher, but this is probably higher than expected for encouraged code sharing. So again, depends on your perspective if this is great news or still room for improvement. A couple of examples of proposed policies that are working as intended or as hoped for, I would say. I think one great example that comes from the Dutch main science foundation, main science funder in the Netherlands. And this was a pilot program that they're starting to report results from. So this initiative called for funding specifically for replication studies, or specifically for funding studies that were replicating previously published research, and ended up funding 24 mostly direct independent replications of previously published research, and the results of those are starting to come out. They have a database of those studies on their website, which is fun to peruse. But they recently came out with some, from their perspective, some summary findings and some conclusions just about the experience of the call for replications, the process of reviewing them, and essentially what they've learned and what they recommend others take from this. So they categorize various the variety of lessons that they glean from this exercise is that it's possible that the research findings are going to corroborate original findings, or in other cases, it's going to uncover unreported details, some of those, we call those hidden moderators, or special cases that weren't reported in the original research findings that are necessary for detecting an effect. And if there's funding to try a third or additional rounds, those can really be teased apart to understand what the main research finding is really pointing to. They emphasize the ability for this exercise as having high educational value. It really helps to articulate to the replicators the necessary details that are needed to include in their own research, because, and this is something that we've learned at COS from doing a variety of replication studies, as you start to get into the details is the point in time when you really understand what is needed to be included. Doing those replications takes patience, and then it improves your own research as you kind of understand better what's actually needed for later replication. And it just highlights the need for improving clarity. And they came out with several recommendations to journals, funders, and universities about why and how they should appreciate the value that replicate. Too often the story is that we already know that. Why would we fund it again? Or why would we, include that in hiring and tenure decisions? Or why would we publish that if it's already been conducted? It's not original. And they have several strong recommendations why these are worth conducting, funding, publishing, etc. All right, and we're going to point to a couple of examples of problems that were found that are being addressed. And so these are examples of meta science in action, I would say. And Hardwick and Ioannidis were able to, several years ago, look at all of the published registered reports that had come out up to that point, and discovered that about a third of them, or nearly half of them, did not have a protocol available for what had been accepted at stage 1 of the registered reports. And so, in fact, they were not living up to their name of actually being registered in any sense of the word. And so Chris Chambers and myself looked into the journal policies of journals at that time that accepted the publishing format of registered reports, and through a lot of back and forth, a majority of them quickly updated their policies to include protocol transparency as being a necessary condition for accepting a register report. And there are a couple of different mechanisms for achieving protocol transparency. They could publish the accepted protocol. They could publish it but embargo it until a certain period of time or until the results are known. Or they could require either themselves to register it or require the authors to register the accepted protocol. And one of the infrastructure improvements that we implemented on the OSF registry is a dedicated template for accepted registered reports, and those are available, of course, on the registry. I'm going to give several examples of good things that are associated with open science practices. So again, under the spirit of good news, some of these are really aligned with what we'd like to see happen. Other good things that are associated with open science, I'm going to point to some citation advantages, which I feel a little awkward pointing out those as being like great side effects of open science, but we all still love, of course, getting recognized and cited for the work that we put out there. So I think that's okay too. One thing that's associated with these open science practices is increased trust and credibility from the public. And so this is a survey conducted of folks in the US from the Pew Research Center. And they found that a strong majority of respondents saw that openly available data underlying empirical research results increases trust to the public from researchers. So there's a variety of things, and it was the one that increased trust most greatly amongst all the things they asked. So that was a pleasure to see, of course. A couple of citation advantages. We see papers that have open data available or associated with them or underlying the results receive a citation bump, and there's about a dozen or so studies I found relatively quickly looking for those, and the quick back of the envelope or back of the napkin calculations. I saw the range from 20% to almost doubling to 100% advantage of papers associated with open data compared to whatever comparison group the authors chose, but similar papers without open data. And somewhat similar findings for papers that had a preprint associated with them. And there are a lot of different mechanisms by which this could happen. Some of the algorithms are combining citations to a preprint with the final published article. There is, of course, more time available for people to find and then cite work when it was preprinted. So there are a couple, as I said, a couple of different mechanisms by which this could happen. But it's in the several papers studies that I found looking at citation advantages of preprints, they all found a bump between 30 and about 60%. I think the last couple of examples I'll show are a combination of things, proposed policies working as intended, and good side effects of open science practices. So Allen and Mellor found that the registered report publishing format is working as intended. So they scored null results from 153 hypotheses from replication and from original studies that are published as registered reports, and found a distinction between, or found a difference between those that were replication or novel hypotheses, and compared those to what's shown in the just general published literature. Only about 5 or 20% of papers published in the regular literature showing up as null hypotheses. Similar findings from Ann Scheel and colleagues looking at the psychology literature, the standard literature versus registered reports, and found somewhat similar findings with about half of published papers as registered reports reported a null finding compared to the vast majority of papers published in the traditional format. Last thing I'll mention are some improvements coming to our main policy recommendations. The Transparency and Openness Promotion Guidelines are a policy framework that we use to advocate for journals, funders, and universities to adopt for increasing open science practices. And those were published in 2015. And there's been a variety of evaluation studies on them over the past several years, looking at enablers and barriers to implementing them, evaluating the author guidelines based on the framework that the top guidelines provides, and looking at what caused integrated reliability issues or problems, and then specific calls for adding in new standards or new practices to top, namely protocol transparency. If you're familiar with the current top guidelines, it is these 8 practices that are can be implemented in one of 3 levels of increasing rigor. But even though this is a summary figure, this is an oversimplification. There are actually several things that are different from the oversimplification. Data citation and increasing design and analysis transparency merely encourages at the first level. The study and analysis plan per registration standard actually flip the verification and requirement levels. So that's inconsistent with others. There are good reasons for that, but I won't get into that because of time limitations. And several of the standards are interdependent. And so these issues, along with consultation from other publishers and implementers, went into a revision of the top framework. And we separated out different parts of top, specific practices, specific verification steps, and specific types of studies, all of which go to increasing the verifiability of an empirical claim, and are proposing and are submitting for publication these practices from registration to reporting guidelines, the increased modularity so that each of these policies can be independently implemented, and at each level can be implemented in an independent manner. The computational reproducibility, which always relied on at least 2 different practices, is pulled out as its own set of programs. So is comprehensive reporting can be completed in a couple of different ways, such as registered reports and a few others. And then finally, several types of initiatives or paper types can be encouraged in a couple of different formats. With that, I would like to thank you all, and we'll be here for questions through the end. Thanks.
All right, great. Well, thanks everyone for having me. And this is a little bit not my usual sort of approach, maybe not even my usual personality. So I'll see how it goes. I fitted myself with a little like electric buzzer, so that, you know, if I start to say anything snarky or critical, I'll just the organizers actually have the button, and it makes it all the way here to New York somehow, and I'll do that, and then you'll see me you know, smile again and say something positive. But hopefully I will have something to say that actually is up to, I guess I in all seriousness, I think in this line of work, and I know that my co-founder and good friend and colleague Adam Marcus at Retraction Watch feels the same way. You really, as sort of cynical and pessimistic as we often are, you have to at your core be a bit of an optimist. Otherwise, it sort of would be so disheartening, and so it would just sort of destroy your soul every day, looking at bad behavior and bad outcomes. I don't know, maybe I'm unique that way, but that would kind of, I wouldn't be able to do that. So my general idea here again today is that there is good news, that in fact retractions being everywhere is good news. Most people, that's at least a counterintuitive idea, and some may even think it's wrong, which I'd welcome that critique. But I think that it's probably because of the sort of stigma that all of this has earned over the years, some of it, you know, well-deserved, but not all of it. And so it's my general idea is that you're hearing lots about all these things, about these bad behavior, about good behavior too, but about retractions in general, and that's a good thing. So let me just start really basically by talking about, by letting you know what a retraction is. Maybe that's very obvious to this audience, but I think it's worth noting that all a retraction means is that a paper, something you're looking at, has been deemed unreliable by, if you will, the powers that be. Now, that could be, generally it's a journal or a publisher, sometimes it's others, but then most of the time, it doesn't actually tell you why the paper was retracted. It doesn't tell you that there was misconduct involved or bad behavior. It just tells you this is this is unreliable. Don't rely on it. Don't cite it. Don't cite it without noting it's been retracted and what have you. I think it's important to level set, even though lots of things flow from retractions and there are lots of other things going on. It's important to at least know what I mean when I say retraction, and that's actually all I mean. So first up, again, retractions are on the rise, and I would argue that's a good thing. I'm not going to belabor the point or this particular graphic, which many of you may be familiar with. It's from Nature from give or take a year ago in December of last year. You'll see a couple of things here, a couple trends. One is the general trend line of retractions growing since 2013. In fact, if you were to go back in time, they're growing, quite steadily since about 2000, 2002. You'll see a big spike in 2023, last year. In fact, that spike grew higher by the end of the year. I'm going to talk about what happened there in a few slides, so bear with me. But, the fact that retractions are on the rise, I don't think is going to be controversial to anyone. I hope that this isn't, but if it is, happy to sort of provide the actual data upon, well, not upon request, is the data available. You can go click on Retraction Watch and find all the data. Crossref acquired those data, and you can do whatever you want with them. But the rate of retraction has grown from about 1 in 5,000 to about 1 in 500. We would argue it probably should be higher, but now I'd have to give myself the buzzer for being a little bit cynical for the moment. But it has risen, and we would argue that this is good news in the sense that science is clearing out the stables, is actually cleaning out the stables. The fact is that we're not at peak retraction. We need to sort of get there, and that would be even better news. But the fact that there are more retractions means people are actually paying attention and doing something about it. The people who are doing that are these people. Some of whom you may be familiar with, some of whom may even be in the audience if I sort of understand who the audience is. And obviously I know where you are, and there are a couple of your fellow country people on that list. And this is not an exclusive, it's not an exhaustive list. It's just sort of where we are right now. We keep adding to it and what have you. But these are the people who are actually finding all the problems. And it's really good news that they're around and doing the work. And it's good news that they're able to do the work, which they really weren't, say, 25 years ago when papers weren't online. You've heard about open science, of course, and open publishing from David. And that's all sort of consistent with this idea that with more eyeballs or mechanical eyeballs or robot eyeballs or whatever it is, looking at what we're looking at, these people can actually do their work, and they find image manipulation, they find plagiarism, they find statistical problems, all sorts of things. And that's great. I'm gonna turn off the buzzer for a second and say the bad news is that a lot of them toil for nothing or next to nothing, and can be threatened with lawsuits. And so I guess if I'm going to flip that around again for good news, I'd say that their growing visibility and their growing prominence and impact has meant that those things are maybe getting eroded. And in fact, there's some support for some of these folks, although I would argue not nearly enough, but still working. And one of the ways that, again, in terms of a positive development, is that a number of journals have actually hired, we didn't write this headline, I don't actually love it. I don't love the word czars, despite the obvious origin of my name. But the important thing, the good news here, is that journals, and at the time we wrote about several, there were a couple that were doing this for even longer before this, but this is 2018. They've actually started hiring people to look at And some of these teams are getting quite large now to look at problems in literature, whether it's before, before something's submitted, after it's submitted, et cetera. And so again, that's good news. And here in the States anyway, and probably everywhere, politicians like to talk about being job creators. And so Adam and I used to joke when this was first happening that in fact we were job creators because people, so I guess that's good news too. If I want to pile on here and talk about there are people who actually have jobs doing this kind of work. So part of the reason that a lot of this is happening is how much attention it's been getting. When Adam and I launched Retraction Watch 14 years ago now, it's not that no one ever wrote about retractions. I mean, they certainly weren't writing about it every day the way we started to do, because we realized it was such a good, sort of fertile ground for stories. But it was pretty episodic. Nowadays, you can't sort of skip a day without reading about some, usually it's sort of couched in a frame of a scandal, but about some, whether it's a retraction, whether it's a group of retractions, whether it's journals being closed. These are just a couple examples. On the left there is my, this is why I was never very good at science, and I certainly wouldn't be a good fraudster, because that's like how I would do photoshopping of a Western blot if you left it to me. So you would know that, in fact, I had faked it by circling that headline because I figured you'd be hard pressed to see what I was talking about. These are both front page stories in, what amounts here in the States to the two largest newspapers. And this is around the world. I mean, wherever you're all from or living, Australia, certainly there's a big story actually picking up on something we did just last week. So, the media is taking note. And again, that's leading to more scrutiny. It's also leading to, especially when it's in the Wall Street Journal or in the business press, it's leading to the markets taking note. Now, frankly, on the day that this happened, when the Wiley stock price dropped 16%, I would imagine that wouldn't be considered good news for stockholders of Wiley. Now, again, and I'm maybe being too Pollyannish, but I would argue that it is good news in the long term, because it means that, again, Hindawi and Wiley, and this, by the way, was what's responsible for that massive spike in 2023. This was over the course of end of 2022 to beginning of 2024, there were 11,600 retractions just from Wiley Hindawi journals, because they had been overrun by paper mills, which I'm going to say, at the risk of offending my hosts, it was not good news that paper mills overran these journals. However, doing something about it, you could argue, is. And so when the markets take note, that also, again, creates some pressure and some, even if it's shaming, and that leads to, if not better behavior, at least scrutiny and more attractions, which again, I think are good news at this point. Governments are also taking note. And this could turn not so wonderful news, depending on who those governments are and who makes up those governments. I say while sitting in the currently still United States of America, as far as I know. But this is, just one example. The House of Commons, I gave some testimony here, and others who are in the audience might have actually as well. The details of this are interesting and worthy of discussion, not worthy of taking time now. But the point is that, a House of Commons, a UK House of Commons signs, a committee said this is worth paying attention to. I would argue that that is going to be good news in the long term. How it might feel in the short term, we'll see. I want to give you, and I'm going to do like really brief, sort of just as some examples of people doing the right thing. I think one of the things that the increase, like not the pressure and the scrutiny, but just the fact that people are talking about these issues has led to, is that people are willing to do the right thing. And so this is you may have heard about something called Pruittgate or Spidergate. Kate Laskowski was one of the people impacted by that. She was the victim of someone who, well, let's just say we don't know where the data came from, and he's no longer in that role. He's Jonathan Pruitt was his name. She went public with this. This is from Retraction Watch, which was actually first. It was a quote from her blog. She went public with this. The community came together around her and others who were victims of this person, Jonathan Pruitt, and said, let's make sure that there isn't, or there's minimal collateral damage. And in fact, there was. And so I would argue it's good news that people are doing the right thing and getting together as a community. there are people, retracting papers, even though it makes them feel, as this quote says, like a fraud. This is the sort of thing that we should champion. We actually have a doing the right thing category, and giving voice to this and telling these stories in a fulsome and clear way, obviously a public way, we think is important. This one is a little more complicated, and they're all actually fairly complicated, but this was a sort of no drama retraction. People said, there's a problem, and then, of course, I used a cute CCBY image of a puppy to maybe make you all smile. It's good news, puppy. It's all together. Things like that are happening. No animosity is a quote again. No animosity between us. This frog paper, which kind of had made someone's career. Well, you know what? It was wrong. It turned out the frog did have lungs, and let's just do the right thing and retract it. Again, this is a story, a sort of narrative we've seen at least a few times, where people, maybe they order the wrong reagent, they order the wrong, in this case, mice. We've seen a couple like that. Again, that you can see how that would be devastating, both from the point of view of wasting time, but more importantly, I mean, these are mice who that we all know at some point were sacrificed. You could have just sort of swept this under the rug, these researchers and others, but they didn't. And they said, there's a forum now for people to talk about these issues. That's a good thing. The really nice thing, if you want to, and I share David's, sense of irony when people like the two of us or any of us today are talking about, citations as a sort of metric of any kind. It's got its problems, but one of the things that economists like to do is look at, again, human behavior. And so they look at a data set like retractions and citations. And it turns out that when you retract a paper for fraud, misconduct, and it says that very clearly, you see a dip in your citations, and like the whole subfield might see this. But if you retract a paper for honest error, and it's clear that that's why you're doing it, and you came forward yourself, you actually don't see a dip in your citations, which, again, with every caveat about flaws in using citations as a metric, does, I think, at least and the economists agree, there's several papers like this. This sort of suggests that at least unconsciously people are rewarding good behavior. We think that's good news. This one's a little bit meta, and you could even argue self-serving, and so be it. If you're going to make me talk in positive language, I'm going to, toot our own horn a little bit. This is a paper, we didn't know they were doing this. We knew they were using the Retraction Watch database, but they actually found that when we covered a retraction on Retraction Watch, people were less likely to cite that paper than if we didn't cover it, which is an argument for us to have 100 times more funding so that we can, cover every retraction and then lead to less waste. But, well, I said it, you can do whatever you want with that information. It's kind of a fun little thing, honestly. But again, this kind of scrutiny actually and awareness actually leads to good outcomes. A couple of other studies that have come out, one of which we were involved in with some students from Duke. And generally these two, and again, you can look these up, and happy to provide, of course, the references. But, one of them said, that we were not involved in this one, said that, in fact, notices and metadata are improving. In other words, you can actually find the information about retractions, which was always a big problem, which I'll get to on my next and last slide, and why we have a sort of solution for that. And the other is that we did this work with some Duke students. They did the work really, and we were, sort of obviously supervising and sort of helping out. But they actually found that retraction notices by a couple key rubrics had improved over time. So that's some good news. And again, if that leads to clearer citations and clearer understanding, that's better. And so my final slide here, I just want to give you some, again, you could argue I guess, self-serving good news. But one of the things that we realized very early on was that retractions were a lot of them were missing from the databases and the indices that people were using. So we launched with some wonderful, generous philanthropic support. We built and then launched the Retraction Watch database. Well, all of this software actually incorporates that the data in order to give you automatic retraction alerts, so when a paper that's in any of the software, in your database in your library has been retracted. You don't have to do anything; it's just automated. But even better, good news is that Crossref acquired the Retraction Watch database a bit over a year ago now. We were so delighted. It actually provides significant financial support for us, and we're able to maintain that part of the work, you know, kind of hopefully forever, because we still do all that work and get a fee for that. But even more importantly, I would argue for the world, is that the data are now all completely free and available, and you can just literally click on a link and get the whole data set in your, you know, in your downloads a second later. And so that we think is good news, and hopefully more people will use it for the kinds of things that I was talking about. That's my contact information, which I think you can probably all figure out anyway. So in the interest of time, I'm gonna stop sharing. And there we go.
Well, how can I possibly follow such great colleagues with some other good news? So I, again, being tasked to talk about some good news, I generally do talk about good news, believe it or not. I do have a conflict of interest statement that I must make clear. I am a co-founder and CEO of SciCrunch Inc., which takes care of SciScore, which I will talk about a little bit later in the slides. So we have heard about several checklists already. There are more, obviously. The US government has a list of these kind of preclinical reporting guidelines, which largely overlap things like Consort, Arrive, and some of the other checklists. The only reason why this one is so important is that every single person who submits a grant to the National Institutes of Health in the US actually is supposed to be following these guidelines. And one of the things that's in these guidelines is, of course, best practices for things like antibodies, cell lines, and animals, which you just heard Ivan talk about. Hey, do I have the right mouse? So taking a step back, you know, why are these kind of key resources, key biological resources, so important to the NIH to put them into their guidelines? This is one example. Here's a lovely paper. The paper is fine. However, there is this reference to a particular research resource, in this case, a mouse anti-flag antibody from a company called Sigma. And if I look for this thing, I have many of these things. And so, of course, one would argue that if you would like to use an antibody, you might want to use the right one, because I personally can't afford 17 of these, either in time or money. They're about $500 a piece, and they take about 6 weeks to figure out whether they're working or not in your lab. So, you know, another fun fact, one of our colleagues at the Non-Human Primate Reagent Resource Center had sent us this paper, which he said, yeah, here's a great paper that actually cites one of our products. And this is the citation. And if you want to look at this, this is an antibody, which is an anti-CD8 antibody. It has an ID, which is great, but it was supplied by Keith Riemann from the National Cell Culture Center. And so what I said to our colleague Diogo, said, hey, did you guys change your name? And he's like, well, no, we're actually never the National Cell Culture Center. And Keith had left. He was the former director about 10 years before. But this is how a relatively recent paper is actually referring to that reagent. So what did we do? We took all of these reagents. We took about 3 million antibodies. We took about nearly 1 million now mice and frogs and rats and all these other critters, worms and fish and other various things. We also took a lot of projects, like in this case, this is a NeuroMab project. And we gave each one of them kind of a landing page with some dedicated metadata. And we said, if you're going to cite this thing, and we, among a lot of our colleagues who run actually these stock centers and repositories, we said, hey, there's going to be some consistent metadata. There's going to be some references to other things that we know about in this space, like the university from where it comes from, which has a ROR ID and other fun things. And then what you can do is actually you can ask authors to actually cite these things using something called a persistent unique identifier. Persistent unique identifier is persistent, right? It's an ID. It's not anything magic. It's just a number, right? But what it does is it's associated in a persistent manner with a set of metadata. And you can update that metadata, for example, if the resource moves from one URL to another, you can actually flip that out, but still it's the same thing. And by the way, when we've looked at core facilities and other resources within universities, they all tend to change all of their URLs, breaking them every roughly 5 years, because somebody in the middle of every university seems to think that it's a really, really great idea to change, university name biology / something to biology / university or something similar, which is really just completely stupid, but whatever, they're going to keep doing it. So if you then use those biology / whatever URLs in papers, they tend to break pretty quickly. In fact, in about 2 years, 25% are broken. So that's why the persistent unique identifier is actually a much better way to do this. So this is just an example. And in this example, there are 1,300 people who have cited this research resource. I'll also show you a little bit in a few slides about ratings and alerts of each of these resources. In this case, this is another research resource. It is a microscope. So we have actually increased the coverage of the RIDs to actually also include instruments. And you might ask, how would someone, how do we get any research resources into the scientific literature at all? Well, it turns out that we've been working with journals for about 10 years now. About 10 years ago, we started a 3-month pilot project. We are currently in its 10th year. No one has ever said that we're going to, continue this for a full project, but we're still in the pilot phase. I'd say we're probably good to go at this point. So what they do is they put us in the instructions to authors and they say, hey, if you've got an antibody or if you've got a cell line or if you've got some other things, go ahead and go to this portal and go search for your research resource, and then find the little cite this button or this cite this line here. Go ahead and copy that, paste it into your paper, and then the paper gets published, and magically you have lots and lots of papers that can now be aggregated based on that ID. If there isn't an RID for your particular resource, we actually give you on the main page a little button that says add a resource. We think it's pretty clear, but we always get questions. Here you can choose your type, and that gets you to the right place where you can actually register your mouse or your core facility or your antibody or other things. And there's no need to be logged in for any of this. You don't have to get an account because that's very annoying. Lots of these resources actually have RIDs now in them in the place where, how do you cite this particular cell line? How do you cite this plasmid? How do you cite this Jackson Labs mouse? The RID is right there. And so they try to make it easy. About 2,000 journals have begun to do this in some different ways. So some of them are just in the instructions to authors. Some of them actually require this. So, thinking about those top guidelines, some of them are just requesting, some of them are requiring, most of them are just requesting or allowing. But in this case, actually, Science and actually Nature, Cell, and some of the other big journals are actually requesting fairly strongly the addition of RIDs, including this checklist for authors. This is the MDAR checklist, the Materials Design Analysis Reporting Checklist, which is supposed to have put together Consort, Arrive, and a bunch of other things, creating this new checklist again. And this is now implemented across and present for every single science family journal. So that's kind of fun. So the question is, did it work? Do all of these interventions sort of work? Can we measure any kind of difference once these things have been implemented? So the nature checklist was an interesting kind of case study that we looked at using some automated tools from SciScore. This is published in iScience now quite a few years ago. But we looked at, for example, a bunch of things like whether organisms or antibodies are identifiable in a particular paper. And what identifiable means in this case is there a catalog number, a stock center number next to that entity, which we recognize as an antibody or an organism. And of course, we've done the same thing. We've looked at, whether the authors are talking about sex as a biological variable, whether they're actually addressing blinding. So some of the rest of those guidelines that came out in 2016 from NIH and came into the Nature Checklist from between 2013 and really centering on 2014. And those are continuing. So the bad news, I would say, is that before this Nature Checklist intervention, Nature was actually very, very low on average in terms of all of these features, including, some around 10% identifiability of animals, around 10% identifiability of antibodies, which, on the bad side would be, 90% of the time when you read a Nature paper, you don't know what they're using. But those things all increased. And as I think David was saying before, there's still a ways to go, but we definitely have seen a tremendous amount of improvement in this. So we looked across antibodies from the 1990s through 2019 here. This is the iScience paper from my student Joe Menke. And what we found here, this is across 300,000 citations of antibodies across all these papers. We see that about 10% of them in the 90s were associated with a catalog number. That's very low. And then that does increase, and it increased relatively slowly to about 20% in 2014, which is where the RID pilot started. And of course, at that time, we didn't have very many journals. We only had 25 enforcing anything. And if you look at the entirety of the literature, as we did in this study, it doesn't really move the bar. But one of the things that happened was that in 2016, we had both eLife and Cell, and actually all of the Cell Press join the RID initiative. And they moved really quickly. So they went from a kind of an average-ish reproducer. So these funky lines here, the two green lines are, Cell and eLife. And when they joined, they actually drove their own identifiability up to about 90+%. And what that starts to do with these big kind of visible journals is it actually brings the entire average up quite a bit. So the average paper had 40+% identifiability of antibodies within about 5 years of starting this initiative, whereas before we had done much, much less over that 4 times the timeline. So I think the ability to improve the overall scientific literature is attainable, but it does take some work from the journals. Here's kind of a similar study looking again at, this is a more recent, this is only a preprint currently. And what we did here is we looked at every single time that a person looked at a particular mouse, and we categorized that into, can I find the mouse? Can I not find the mouse? And if I can't find the mouse, I'm going to call that this in this orange category, which we called those nicknames. This is an example of a sentence. Jackson Laboratory, and the Spry 2FF mouse from MMRC. Now we know the stock center is MMRC. We know this is the name of the mouse, but if you search this thing, you're not going to get back to a single mouse. This is an example of another mouse, which is more of the full name. So we found this mouse. This is actually a full name of a mouse, or very close to it. But you can see that this is a very, very long name. And so most people just call it the 5X FAD mouse, which is fine. But if you just use 5X FAD, then you will get multiple mice. So if they're using the full thing, then you can get this. And catalog number can be provided. So catalog numbers are great. They're findable. Your colleagues can figure these things out, but they're not machine readable. RIDs, this is an example of an RID syntax in this paper. And again, this is another 5FAD mouse, and that is the machine readable aspect of it. So what you can see is that half of the time in 2011, the mice were not findable. We couldn't figure out which ones they were nicknamed. But in 2014, when we start getting just a few papers in the scientific literature with RIDs, we are starting to get a slight decrease in the number of these nickname mice. And then in 2016, when the Stock Center really went after their authors and emailed all of them, I think ad nauseum, at least 10 times that particular year, we're starting to get many more of these mice being cited using the RID. But the most important part is you can see these nicknames, they're almost being eliminated. We're down to about 5% nicknames in the literature now for the MMRC mice, which is, I think, amazing, going from 50% of their literature down to 5%. This is, I think, really a huge gain. This is a huge gain for reproducibility. But it involved absolute work from the Stock Center. It involved work from the journals, but also really the Stock Center and those authors. So one of the things I like to talk about is product recalls. Now, scientific products, believe it or not, have no robust way of being recalled. If you have an antibody in your lab, it turns out you largely will not know whether that antibody is not doing what it's supposed to be doing. If the cell line is overgrown by another cell line, you will not know that. But it turns out that the RID system, because it forces the author to go into a single database and ask the question, give me my RID, I guess I need to go look this up, oh darn, right? What it does is if it has that kind of recall information in there, so in this case, the 15C4 cell line has been labeled as a problematic cell line. It's been labeled contaminated by one of the projects that was led by Amanda Capes Davis from down under, probably near you guys somewhere. What this cell line is it is a poor cell line. It should not be used. And authors who are in the final throes of the publishing process, when they see that, they've already done the work. They've already done all of this stuff. But what it looks like to us when we looked across the scientific literature is that even at that stage, when they are asked to look and put this into their paper via RID, they don't. They look at these warnings and they heed them. They have yanked papers, they've yanked figures, they have redone things because they don't want to stand behind something that isn't a good quality set of research. So what we found here is again, a text mining study that looked at over 150,000 papers. All of these were basically the selection criteria was they were all open access papers that were text mining accessible. And these papers were found to have at least one cell line associated with them. And when we looked at those papers, 16% of them were actually associated with a cell line somewhere on the register of the ICLAC, problematic list. We didn't look at each one of those because it was way too much data. But at the time, we had roughly, the same thing, but roughly 5% of these papers that had RIDs associated had a cell line on on the problematic list. And because that was a smaller number, there was only about 700 papers to look at. We looked at those 700, we looked at the ones that were problematic, and we noticed that there was only one case where an author had seen something like this and still continued to put that into their paper. So 1 out of the 50. The other 50 had, were things like HepG2 cell lines, which are perfectly fine liver cancer cell lines, and they were being used for something like liver cancer instead of hepatoblastoma, which is what the incorrect labeling was on that particular cell. So there's the ability to intervene with authors actually produces decreases the poor practices, and those poor practices are because of not remembering to look something up in the ICLAC register, as opposed to wanting to do a bad job. So using these identifiers actually does make papers better. It makes them more reproducible. We have lots of warnings that are starting to come in. We have various large and small projects that are actually contributing data to the Resource Watch. Sorry, Ivan, you probably have that trademarked. But in any case, we have projects like ENCODE, Icaros, lots and lots of core facilities, core shared facilities are actually giving us data. With software tools, we've seen some software tools that have had reported problems where, they're giving the wrong answer under certain circumstances. Some animals don't recapitulate a disease, and you might want to know that before you stake your reputation on this kind of thing. So one of the things that we've often struggled with, however, whenever you have these guidelines, just like the first speaker was talking about, how can we make this actually easier for authors, for reviewers, for editors to actually implement? And our decision was to get funding from our government to build something called SciScore.com. This is an example of a report that we now produce for every one of the authors in a journal that is participating in the SciScore.com practices. And what they do is they put their method section into their regular submission system, and then that goes in a very kind of simple way to our system. It pings back to the author section, and they get back a report. And this is an example of a report. There's a nice summary with lots of how to improve the paper kind of things, but also just a summary. And then here are the other things that it does actually show. So for example, in antibodies, here's the author's sentence. Here is a recognized antibody. And then here is the RID that's associated with that. And how to improve the paper? You're good to go. You don't need to improve a thing. But in this case, we know that this protocol is not resolving properly. So this URL to protocols.io seems to be broken. And so there's a bit of a text here that tells you, hey, you might want to, check that. In cases where we do detect statistical tests, we try to get people the information about, for example, the kinds of things they should be reporting about every statistical test listed in one relatively easy place. trying to get them to understand what the assumptions are for each test that we recognize. So this actually, we think, will change the way that the way that we're actually implementing these policies. We know that there is a rise in all of the things that we're looking at over 20 years. So this is the average SciScore. So there's that average number, but things like, for example, sex is a biological variable is improving. Randomization reporting is improving over 20 years. Blinding and all these other checklist items are also improving. But for journals like the American Association for Cancer Research, which really pushes both RIDs for reagents, and they're doing it by using SciScore, you see that improvement is a whole lot faster. So in 2019, we had about 3% compliance with RIDs, and currently we have about 43% compliance with RIDs in the journal of or in cancer research. And just kind of a fun one, I threw this one in. What are you looking at? So PLOS One had actually always been very into making sure that your code is cited. And so we kind of did this little experiment with cancer research. This is one of the AACR journals versus PLOS One. So we see the total number in the kind of dark bars of papers that have at least one mention of GitHub. And then there's the percentage, which is in the lighter colors of articles with GitHub. And one of the cool things that we found is that, okay, while PLOS One definitely has many, many more papers per year mentioning GitHub, if you look at the percentage, they're about, between 3 and about 5% of the total number of papers. They're actually talking about GitHub, which is sort of a proxy for code sharing. But if you look at cancer research, I mean, come on, these are not coders, right? But if you look at them from 2018, they're, not doing as well as PLOS One. Certainly, the total number of papers is very small in that. But as a percentage, this tends to grow. And with the introduction of SciScore, just giving people those little nudges, it seems to be growing as a percentage. So we've really overtaken in 2023 PLOS One. In terms of code sharing, if you can look at this is 15% of all of the papers in cancer research are actually reporting their GitHub repositories, which I think is really cool. So thank you very much for listening to me talk about RIDs. And I would like to just encourage everyone in the room to please be a joiner. Put RIDs into your next paper. It might save you from a retraction later. And if you have any concerns about a resource, please let us know. There is a little link on the bottom of every one of those where you can submit your problem or just contact me. Thank you. I should probably stop sharing, huh? Are there any questions?
Thank you, Anita, for all that good news. We have a few questions in the Q&A. So from Paulie, a question for our first speaker, Tammy. The question reads, great work, Tammy. I wonder if, given that reducing research waste is now in the Declaration of Helsinki, whether there is an appetite now to integrate checklists such as TIDIER into ethics applications.
Last month, and certainly.
Oh, yeah, come up here.
No, Paul can't hear me. Thanks, Paul. I said that new declaration only came out last month, and I guess speaking from the Equator Network perspective, we were very excited to see that reporting guidelines are now mentioned in it. So I think there's certainly, that might be the impetus we need to get a little bit more serious use and checking of reporting guidelines. So something to work on.
Any questions from the floor here? Anita.
So if you were going to predict where the next good news is going to come from, where would that be? Who's going to break? Is it we're going to get some great news from funders, from the media, from researchers, from publishers? Where's it going to happen?
Can we put it out there that MRFF's going to put targeted calls out for meta research? Can we like put it out there and hope that someone
I didn't hear that.
I'm just putting it out into the universe.
Oh,
and
yeah,
perhaps
okay, yeah.
someone with
Yeah.
power will hear. So we have a national funding scheme that does targeted calls for priority research, and it's nearly always clinical. And it'd be nice if it did meta research, wouldn't it, Adrian?
For our speakers, David, want to chime in at all? Remote speakers. David, can you hear you?
I'll put something out there. Yeah, I think, and I hope funders and others will kind of encourage or consistently fund replication and direct replication research, because a lot of the research on that is taking snapshots and just kind of one-offs of individual studies or a group of studies here and there. And if we want to see kind of trends in that issue, we want to be able to look at it over time and see how various activities that the meta-scientific community is looking at are affecting rates of replicability or the barriers to do that. And so I expect that would be my next hopeful good news story to share.
Any questions? Anna?
I'll do that thing where it's not really a question, sorry. One of the things that strikes me.
I can try to repeat it, or you can come up.
Can you repeat it for me? One of the things that strikes me about these talks is that there's an overarching theme of being connected to something that already exists and using that as a stepping point to getting people to actually change things. And in the spirit of your question, Alex, is there something like that in the grant world that we could be linking in with?
Hannah points out that a lot of the talks reflected that the improvements came through linking into something that already exists, reporting guideline, consort, and Retraction Watch now is a longstanding institution. Is there something in the grant funding side that we could link into to try to get tools more widely adopted or get reporting improved, et cetera? I know that ethics is another common thing that often things connect to. Anita?
Well, we do have, I mean, so at least on the US side, and I think that there are similar things going on in Europe, and whether or not you consider England Europe or not, that's entirely up to you. But there are some things going on in Europe that are relatively similar. I mean, we've had a real push from the at least the National Institutes of Health over on the US side to improve rigor and transparency of scientific studies. I know that similar things are happening in various parts of Europe. I think people are looking. The big scandals coming out of the Alzheimer's literature, I mean, that has made the congressional record. So I think, as much as we hate to be driven by scandals, they are at least a force that probably allows all of us to kind of look at these important issues that maybe wouldn't get as much play otherwise. I mean, one of the universities that we work with is the University of Luxembourg. They try to run, a whole series of tools on top of all of their papers that come out. And the reason they do that is because there was a big scandal with their education minister. And, again, having scandals drive what you do is probably not good, but at least it shines a good light on that.
And we will have to go to the break in a minute, but we've, I've updated, your hand is newly raised, but also Ivan has his hand raised. David, did you raise your hand a second time?
Yeah, I'll just say that along with what Anita said, the U.S. federal government is moving forward with a lot of data availability statements being required for any grant applications. And one of our areas of advocacy is to encourage that those be publicly available. And that would become a sort of de facto registry of all funded work, and would be an area to link all outputs from. So we see opportunities there for improving transparency from the get-go, from the decision to fund. Ivan?
Yeah, I guess I would just say, and I'm not sure it's exactly an answer to that question, but something that always strikes me is ensuring that different scientific communities learn from one another's, whether you want to call them mistakes, lessons, scandals. I mean, we report on scandals all the time. You know, and I don't know, I can't see the audience well enough, but if John Loadsman is there, but raise your hand if you're there, John.
No, he's not here. He's not here. He is not.
So, and the reason I mentioned John is that anesthesiology is a field that very few people actually think about, maybe for good reason, but it was really, even before we look at, you know, psychology and cancer biology, it actually had its sort of taking itself to the woodshed moment back in like starting in 2008, 2009. And the reason I mention that is because they learned a lot and actually were able to accomplish a lot that I think other fields, you know, could benefit from. And so we see from our very bird's eye view, you know, this field, and then everyone goes, oh my God, something terrible is happening. And then everybody reinvents the wheel and all these sorts of things happen. There's a meta problem there, I suppose. But I would sort of encourage people as much as possible to, again, it's not that everything's transferable, but to understand that, you know, someone might have thought of this before. And I guess that's how I'm getting back to the question, which, again, if I understood it, or at least there's a theme there of, oh, there's existing things. Let's link into those. And I would say, let's link into existing knowledge that may be very far from your field, but in fact is very related.